Welcome to the webinar on Drug Design and Molecular Modeling organized by Al-Azhar College of Pharmacy, Thodupuzha. Today's speaker is Dr. Cinu Thomas A., Professor, Department of Pharmaceutical Sciences, Cheruvandur, Kottayam, Kerala.
Okay, as we all know, drugs are essential for the prevention and treatment of disease. And nowadays, human life is constantly threatened by many diseases, previously cancer, and now most importantly, the COVID-19. Therefore, ideal drugs are always in great demand. To meet the challenges of developing ideal drugs, an efficient method of drug development is always demanding. And the process of drug development, we all know the drug discovery process is time consuming, expensive, and it requires the knowledge of many aspects. To fulfill all these challenges, several multidisciplinary approaches are required for the process of the complete drug discovery. And collectively, these approaches would form the basis for rational drug design. So let's see, what is designing? We all know designing is creating something new. And designer is a person who plans the look or working of something prior to it being made by preparing sometimes some drawings or by preparing some plans. We all know that dress design. We design dresses. It is an art of application of design and aesthetics of or natural beauty.
Accessories. What an architect does. Usually he plans, designs and oversees the construction of buildings. Similarly, drug designer. Who is a drug designer? A medicinal chemist should be a drug designer. So, a drug designer designs tailor-made compounds. He aims at developing a drug with high degree of chemotherapeutic index and specific action. It essentially involves the study of biodynamics of a drug besides the interaction between drug molecules and molecules composing the biological objects.
So, drug design is simply called as rational drug design or simply rational design. It is the inventive process of finding new medications based on the knowledge of biological target. And drug design involves the design of small molecules that are complementary in shape and charge to the biomolecular target with which the drug interact and bind to the target for producing the biological effect. Traditionally, the drug discovery was carried out simply by chemical modification. That is, an analog of a known active compound was synthesized with a minor modification. Now also in many labs and academic institutions, only this is happening. That is a traditional method of drug design. They modify an analog of an active known drug, and that may lead to improved biological activity. Example of such a drug is cimetidine. That ranitidine was produced from the structure of cimetidine. So the limitation was that it also ends up with something very similar to what we started with. Some drugs which is very much similar to the initial drug. And so traditional methods of drug discovery rely on the trial and error testing of chemical substances on cultured cells or animals and matching the apparent effects to treatments. But rational drug design begins with a hypothesis that modulation of a specific biological target may have therapeutic value. Hence, the traditional drug discovery processes were irrational and it is based on serendipity and initiation. And mostly trial and error approach was followed in all the institutions. And we know it was time consuming with very low through output. Mostly it was only me to approach. There was no de novo design. So the completion of the Human Genome Project and the development of proteomics, structural genomics revolution, and the development of information technology, all are fueling an even greater opportunity for drug design to be part of the success story in the discovery of new drug leads. So this drug design, what we have told till now, it is the inventive process of finding new medications based on the knowledge of a biological target. So in this scenario of COVID-19, it is very much important. We should know how we can design a new drug for the diseases, newly evolving diseases. Even though everyone is looking for new vaccines, we can design new drugs also. So it is an inventive process of finding new medications if we know the structure of a biological target. And from the discussion so far, we can define this drug design like this. It involves the design of molecules that are complementary in shape and charge to the biomolecular target with which they interact by binding to it. And the drug design process, before starting the drug design process, we should keep in mind the designed drug should have some druggability characteristics. They should have drug-like properties. So before starting any drug design projects, we should keep in mind the rule of 5. That is, the probability of the effectiveness of the drug can be enhanced by applying this rule of 5 or Lipinski rule. It is also known as Pfizer's rule of 5 or simply rule of 5. And it is a rule of thumb to evaluate the drug likeness or determine if a chemical compound with a certain pharmacological or biological activity has properties that would make it more likely an active, orally active drug for humans. And this rule was formulated by Christopher Lipinski in 1997, based on the observation that most medication drugs are relatively small lipophilic molecules. And this rule says that is, all the drugs should have octanol water partition coefficients less than or equal to 5. The molecular weight should be less than or equal to 500. Number of hydrogen bond acceptors should be less than or equal to 10. Number of hydrogen bond donors should be less than or equal to 5. And if 2 or more of these rules are violated, the compound might have problems with oral bioavailability. So we should keep in mind this rule of 5 before starting any drug design projects. But we know all the rules will have always exception. We cannot include antibiotics, some antibacterials and some antimicrobials in this with this rule of 5. So candidate drugs that conform to the rule of 5 tend to have lower attrition rates during clinical trials, and hence they have an increased chance of reaching the market. With the help of computers, we do now drug design. So computer-aided drug design represents computational methods and resources that are used to facilitate the design and discovery of new therapeutic solutions. Drug design with the help of computers may be used at any stages of the drug discovery process. here we are not going to discuss the complete drug discovery process because it starts from target identification, lead identification, hit to lead optimization. So many steps are there in drug discovery process. Drug design is one of the steps during the drug discovery, and this drug design process can be applied at any stages of drug discovery, like hit identification using virtual screening. That is, that time we use structure or ligand based design. Hit to lead optimization or affinity and selectivity. That is, at that time we go for structure based design or QACR. Then lead optimization, that is optimization of other pharmaceutical properties by maintaining affinity. So this drug design, that is when a ligand molecule interacts with a protein, the protein bounds with the drug and undergoes various degrees of conformational changes to accommodate the drug. As a result, the biological activity regulated by the protein is modified. So, the drug design can be broadly classified into two types: ligand-based drug design and structure-based drug design. That is, depending upon the structure of protein, whether we know the structure of the protein, whether we know the structure of the ligands, depending upon that, the drug design can be completely divided into two types, that is ligand-based drug design and structure-based drug design. In ligand-based drug design, we don't know the structure of the target or the structure of the protein or the structure of the receptor. The structure of the receptor is unknown when we go for ligand-based drug design. Whereas structure-based drug design can be done only when we know the structure of the target or the structure of the receptor. Okay, and this is again subdivided into many types. So let us see one by one. That is ligand-based drug design and structure-based drug design. Okay, first of all, ligand-based drug design. Okay, so we have seen the drug design aspect is broadly divided into two classes: structure-based drug design and ligand-based drug design. In structure-based drug design, we know the receptor or we know the target. That is, I am telling the structure of the target or the structure of the receptor. In ligand-based drug design, we don't know the structure of the target. We only know the already existing drug molecules or already existing ligands. Based on that, we design newer molecules for the therapy. Okay, so it is also known as indirect drug design. And the other molecules may be used to derive a pharmacophore model that defines the minimum necessary structural characteristics a molecule must possess in order to bind the target. Okay, so the ligand-based drug design can be roughly classified into two, like pharmacophore mapping and QACR techniques. Okay, pharmacophore mapping and QACR techniques. Okay, so if we know some of the molecules or if we know the structure of some of drugs already existing, we can design newer molecules based on the information of the already existing ligands. That is, these molecules will have certain pharmacophore, and a pharmacophore model can be derived which defines the minimum necessary structural characteristics a molecule must possess in order to bind to the target. Or in other words, a model of the biological target can be built based on the knowledge of what binds to it. And this model in turn may be used to design newer molecules that interact with the target. That is the basis of ligand-based drug design. And it is used in the absence of determined 3D structure for the receptor. And ligand-based drug design can be done by three methods. First of all, we may do random screening. and pharmacophore mapping and QSAR. These are the three techniques when we go for ligand-based drug design. This time we don't know the structure of the or the 3D structure of the target or the 3D structure of the protein or the receptor. That time we go for ligand-based drug design because we only know existing molecules which are used to treat that disease or which are acting on that receptor. So first of all, random screening. When no idea about the receptor and ligand are known, we can search either in databases or in the environment. And databases and combinatorial libraries contains information of new molecules and compounds which are already existing for the treatment. And environment. Environment means the molecules obtained from the natural products, and these can be modified with the help of computers to produce potential lead compounds. And so we do random screening either by docking. Virtual screening can be done on random molecules by selecting from any databases, and we can go for newer molecules. Rarely we use this random screening methods. So the next method, that is at this juncture, I want to tell you what is homology modeling. Homology modeling, we go for ligands. We know some of the ligands and we don't know the structure of the target. At that time, we go for ligand-based drug design. But when we don't know, like this situation, we don't know the structure of the target, we don't know the structure of the receptor, we don't know anything about the pathogen, and we don't have any existing molecules. Okay, so we are completely bind. That time we go for homology modeling. It is also known as comparative modeling of protein, and it is a technique which allows to construct an unknown atomic resolution model of the target protein. So the protein structure is not known to us. The 3D structure is not available. At that time, this technique, homology modeling, allows to construct the unknown atomic resolution model of the protein. And for that, we need its amino acid sequence and an experimental 3D structure of a related homologous protein. We call it as a template. So homology modeling can be done when we have the amino acid sequence, or we say the primary structure of the protein. So it's amino acid sequence, an experimental 3D structure of a related protein or related homologous protein, and that homologous protein is known as the template. And if similarity between this protein under investigation and the template protein is detected, we can assume structural similarity. Okay, if there is any similarity between the sequence, amino acid sequence of the protein under investigation and the homologous protein, if there is any similarity, then structural similarity can be assumed. We know the structure of a protein. It starts from primary, secondary, tertiary, okay, quaternary structure. So structural similarity can be assumed if there is any similarity between the target protein and the homologous protein. Usually 30% sequence identity is required to generate a useful model of the protein under investigation by homology modeling. And then we predict the three-dimensional structure of a given protein sequence. Okay, this is homology modeling. So I started with ligand-based drug design. In ligand-based drug design, we don't know the structure of the target. We don't know the structure of the receptor, but we know the structure of the ligand. And so before going to other models, I told you about homology modeling because I was discussing this because homology modeling can be used when both the structure of the receptor and the structure of the ligands are unknown. That time we can go for homology modeling. So initially we produce a structure of the protein under investigation, and depending upon the structure of the protein investigation, we design molecules for that protein. That is homology modeling. The steps involved in homology modeling are this. That is, first identify the related structures or the templates, the structure of a homologous protein, then select template, then align the target sequence with the template structures, then build a model for that target from the amino acid sequence primary structure and evaluate that model. If the model is okay, then we can use for further drug design. So that is about homology modeling, because every time in drug design we will come to know, or we will come to listen about some talks and all about homology modeling. So this is a just brief idea about the homology modeling. And next is pharmacophore mapping. This is also a ligand-based drug design technique. The basic steps involved in pharmacophore mapping. I told you what is a pharmacophore. It is a crucial parts of a drug molecule that interact with the receptor, and that parts or that functional groups are responsible for the molecule's biological activity. And the typical pharmacological features or pharmacophoric features are mainly hydrogen bond donors and acceptors, positive and negatively charged groups, hydrophobic regions, hydrophilic regions. All these are the typical pharmacophoric regions of a molecules. And in a homologous series of drug molecules or in a series of drug molecules, there can be a common pharmacophore. So first, we have to identify the pharmacophoric regions in the ligands or the drug molecules already exist. And determination of the 3D pharmacophores is known as the pharmacophore mapping. That is the determination of the shape and position of this pharmacophores in the 3D space is known as 3D pharmacophore mapping or pharmacophore mapping. So by doing pharmacophore mapping, or by knowing what are the pharmacophores and where all these pharmacophores are situated, or by doing this pharmacophore mapping, we can go for newer molecules with the same functional groups or similar functional group or analogues of the similar functional groups in the same position, and we can improve the biological activity, or we can decrease some side effects or some adverse effects of the drugs. So this is an example of a molecule where the pharmacophore hypothesis is marked on the active molecules. This pharmacophore models represents features in a specific 3D pattern, and these features are used as a model to screen molecules in any library of compound. And if a molecule, you can see many spheres, and these spheres represent the pharmacophoric regions. You can see here it is written ring aromatics, hydrophobic regions, negative ionizable bonds, then hydrogen bond acceptors, hydrogen bonds, aliphatic region. So these pharmacophoric features should be there in these positions. So we can design newer molecules with the same pharmacophoric features in the same positions. And this is known as pharmacophore mapping. If a molecule can be fitted inside these spheres, and we will consider that molecule as a hip molecule. And this pharmacophore mapping involves knowledge of other molecules that bind to the biological target of interest. And these molecules are used to derive this pharmacophore model, which in turn is used to design newer molecular entities. So then we can build new molecules with increased desired properties using this pharmacophore mapping. And practically, we go for, we do this pharmacophore based drug design when we know the structure of many ligands already existing, okay, but we don't know the structure of the receptor. We have many ligands already used for the treatment, and depending upon the pharmacophoric features of that ligands, we map the pharmacophoric positions, and depending upon that mapping, we design newer molecules which have more chemotherapeutic index. So this is again an example of pharmacophore based drug design. That is, examine the features of inactive molecules and the features of active small molecules, and then depending upon that, we generate a hypothesis about what chemical groups on the ligands are necessary for the biological function and what chemical groups are not required, which chemical groups are required and which chemical groups are not required, or which chemical groups suppress the biological function. Then depending upon that information, we generate new ligands which have the same necessary chemical groups in the 3D location. That is why we call it as it mimic the active groups. The advantage is that whenever we don't know the structure of the biological target, we can go for pharmacophore based drug design. These are the steps involved in pharmacophore generation process in practical. When we go for a software-based pharmacophore generation process, these are the important 5 steps. That is training set selection. That is the selection of molecules already exist. Then from that molecules, we select features or the pharmacophoric features which are required for the biological activity. Then confirmation generation. Then common feature alignments, and finally validation of the pharmacophore regions. This is the processes which we do in computers or in softwares when we develop the pharmacophore mapped hypothesis. So that was about pharmacophore mapping. Then the next method in ligand-based drug design is QSAR. We all know what is QSAR, quantitative structure-activity relationship. This is a correlation between the calculated properties of molecules and their experimentally determined biological activity. And it is a process by which the structure activity and the structure property is quantitatively correlated with the biological activity. And this QSAR attempts to identify and quantify the physicochemical properties of a drug and check whether these properties has an effect on the drug's biological activity by the use of a mathematical equation or statistical analysis. And this QSAR relationships in turn may be used to derive compounds, newer analogues with improved biological activity. So QSAR is a statistical approach that attempts to correlate the physicochemical properties of drug molecules with the biological activity. Here also, we don't know the structure of the receptor. Okay, we only know the structure of some ligands or the drug molecules. So at that time, we calculate the physicochemical properties of that drug molecules, and we try to relate or correlate the physicochemical properties of that drug molecule with the biological activity. And depending upon that correlation, we design newer molecules with improved biological activity. So it is a statistical approach that attempts to relate physical and chemical properties of molecules to their biological activities. And the molecular properties, I told you physicochemical properties. So the molecular properties used in QSAR equation are called descriptors. And a descriptor can be any number that describes the molecules. Okay, a descriptor can be any number which describes the molecule. And various descriptors commonly used in drug design techniques are molecular weight, number of rotatable bonds, log P, hydrogen bond, number of hydrogen bond donors, number of hydrogen bond acceptors, refractive index, polar surface area, molar volume. Many are there to describe each molecule, or all these properties or are used as descriptors in QACR. And many QACR approaches are in practice based on these data dimensions. That is, the descriptive dimensions. Based on the descriptive dimensions, there are many QACR approaches, and it ranges from 1D QACR to 6D QACR. Okay, 1D QACR to 6D QACR, but mostly used techniques are 2D and 3D QACR. Okay, the based on the dimensions of the molecular descriptors, we classify these QACR techniques to 1D to 6D QACR. And I told you the most importantly used QACR techniques is 2D and 3D QACR. The 2D QACR study is mostly based on specialized molecular fragments that constitute the chemical compound. Mostly there are different descriptors that include constitutional descriptors, for example, molecular weight, number of atoms present in the molecule, number of hydrogen atoms present in the molecule, number of carbon atoms present in the molecule. All these are used as constitutional descriptors in 2D QACR. And topology or shape-based descriptors, then total polar surface area, then electrostatic and quantum chemical descriptors, geometrical and molecular fingerprints. All these are used in 2D QACR as descriptors. Whereas in 3D QACR approach, it is a little more advanced, well-established, and the most exploited technique nowadays for structure activity relationship. And this 3D QACR concept is focused on the prediction of biological activities based on 3D properties of the lead compounds. And it uses a series of linear and non-linear analytical procedure or predictive methods. So I cannot describe all these in this 1 hour. If anybody wants to know more about all these techniques, you can contact me. So this is a schematic overview of QACR process. I told you, we already have many molecular structures, which is we know that these molecules will bound to the protein of interest or protein under investigation. Okay, molecular structures we select, or the ligand structures we select, and we calculate the molecular descriptors. I told you, constitutional descriptors, electronic descriptors, geometrical descriptors, hydrophobic, lipophilicity, solubility, steric, quantum chemical, topological. All these are descriptors. All these descriptors of these molecules are quantified, and we calculate the biological activity also. And all these are pre-processes for normalization, standardization, and all other. And all these can be done with the use of softwares, and statistical procedures are done. And then we get a statistical equation. And that statistical equation is evaluated based on these values. And based on that equation, we can predict activity of newly designed molecules. And this is the schematic overview of a QACR process. And this is a typical QACR equation. A typical QACR equation is this. You can see log 1 by C. This is the biological activity. Okay, but we know what is C. C is the concentration, molar concentration that causes a certain biological effect, that is indirectly related to the biological activity, or we correlate the molar concentration that causes a certain biological effect to the biological activity of that compound. Okay, so this is the equation we get after a complete QACR analysis. Okay, and 1.15 into plus or minus 0.2, you can see 1.15 is the regression coefficient, and this is the confidence interval, and pi. Here pi is the descriptor used, that is the lipophilicity parameter, or we can say the partition coefficient parameter. And this is again another descriptor. Here it is the regression coefficient, again confidence interval, and sigma. Sigma is the electronic parameter. So like that, any number of parameters can be included in this equation. And by the statistical evaluation of this equation, we get some values, statistical values, where n is the number of compounds used, that is 22, number of compounds used to train, or the number of ligands where we have used for generating this QSAR equation, and R is the regression coefficient, and S is the standard deviation, F is the Fisher value, and again this Q is a cross validation coefficient, and squared for cross validation regression coefficient is S plus. All these values exhibit, all these values represent the authenticity of the generated QSAR equation. And this time, I would like to tell you a little more about 3D QSAR, because 3D QSAR is the most used technique for the QSAR methodology or the ligand-based drug design. 3D QSAR is a technique used for quantitatively predicting the interaction between a molecule and the active site of a specific target. The great advantage of 3D QSAR is that it is not at all necessary to know what the active molecule or active site of the target, that is a binding site of the target, looks like. Thus, it is possible to use this technique when the target is unknown. Okay, and the first step in doing 3D QSAR is selecting a training set of molecule. This is a set of compounds with known activities, known biological activities. Okay, a set of compounds with calculated biological activities. Ideally, there should be 15 to 20 compounds or more in the training set. Preferably, all should not be on the same backbone. There should be a wide range of activity as possible. For example, the value range of activity is from 1 to 10. We should have all types of molecules from activity 1 to 10, okay, within the training set. And the next step is the generation of shapes and alignments of the training set of compounds. Then select the dominant features which are required for the biological activity, and create the model, and finally validation of the model. That is, after performing the ligand alignment, the researcher can examine for the dominant features to determine which fields might be most crucial for the particular target for eliciting the biological activity. And the model is created from a large curve fit to find a fitting between the fields and biological activity. And this is an automated process that is usually done with the statistical algorithm known as partial least squares, PLS algorithm. And there are different types of 3D QSAR techniques. The most established 3D QSAR technique is known as ChemTalks, comparative molecular field analysis, or ChemTalks. And the development of ChemTalks is based on the concept that biological activity of a molecule is sensitive to spatially localized differences in molecular field intensity. Basic concept in ChemTalks analysis. That is, the biological activity of a molecule is sensitive, that is variable to according to the spatially localized differences in molecular field intensities, and it mostly focuses on the ligand properties, particularly steric and electrostatic field properties, and ligand receptor interactions like favorable and unfavorable receptor ligand interactions. It is an alignment dependent descriptor method. Here also, all ligands, aligned ligands are placed in an energy grid. By placing the probe at each lattice points, energy is calculated, and the resultant energy calculated at each unit or each lattice point corresponds to electrostatic and steric values. And these values are called as or serves as descriptors for further analysis. And these values are further correlated with the biological activities using the linear regression method, like I told you, partial least square or PLS algorithm. So this is the result of COMFA analysis, what we get from the software. From here, we can identify favorable electrostatic field and unfavorable electrostatic field. That is, the blue regions represents the electropositive regions and red is the electronegative regions, favorable and unfavorable. Okay, here the green region is favorable steric field and the yellow region is the unfavorable steric field. That is, larger or bulk groups can be positioned in this favorable region, whereas bulk functional groups cannot be placed in this unfavorable regions. So that's about COMFA. And the next method is COMSIA. COMSIA, or Comparative Molecular Similarity Indices Analysis, or COMSIA. It is a recent modification of COMFA. The approaches of this COMFA and COMSIA are similar, except for molecular similarity, which is calculated additionally in COMSIA studies. COMFA mostly focuses on the alignment of molecules. And so sometimes it may lead to errors in alignment sensitivity and interpretation of electrostatic and steric potential. To avoid all these errors, Gaussian potentials are used in COMSIA. That is, they are much softer than COMSIA functions. The regular energy grid box is constructed as that of COMFA, and similar probes are placed throughout the grid lattice. The calculation is mostly done on steric, electrostatic, hydrophobic, and hydrogen bonding properties. All these properties are calculated at regular spacings of grid points corresponding to a particular descriptor. And these descriptors are important in correlation with the biological activities in COMSIA. Okay, so till now we were discussing about ligand-based drug design. Okay, we can recall all like ligand-based drug design. The structure of the receptor is not known. We know some of the ligands, some of the structures of the ligands. That time we go for ligand-based drug design. In ligand-based drug design, I talked about pharmacophore mapping and QSAR. In QSAR itself, more about 3D QSAR, COMFA, and COMSIA. And in between, I told you about homology modeling also, which can be used when both the structure of the target and the structure of the ligand is unknown. Okay. Now we move on to structure-based drug design. Okay. In the structure-based drug design is the preferred method of drug design because it has the highest success rate. The success is due to the fact that this process gives researchers a clear understanding of how the drug works and how to improve activity, specificity, pharmacokinetics, everything. So the scientist will come to know everything about the drug receptor interaction. It is also known as direct design, and it is used when the spatial structure or the 3D structure of the target is known. Using the information of ligand receptor interaction, we can postulate ligand improvement. Or using the information of that drug receptor interaction, we can design improved newer molecules. For example, if the structure of the active site is known, the steric complementarity of the ligand could be improved to increase the affinity for its receptors. Okay, the structure of the active site is known, or the binding site is known. So we know what are the features required for complementarity of the drug molecule. Okay, the complementarity of the drug molecule with the structure of the receptor site or the active site or the binding site. With the knowledge of these two, we can improve the affinity by modifying this drug molecule for its receptors. Okay, SBDD, that is structure-based drug design, relies on knowledge of the three-dimensional structure of the biological target, a known 3D structure of a target which binds to its natural ligand of a drug or a drug is determined either by X-ray crystallography or NMR to identify its binding site. I told you the active site. And for a lead discovery, that is the starting point of structure-guided drug design for a known target, that is identification of the binding site. If the structure of the active site is known, then the steric complementarity of the ligand could be improved to increase the affinity for its receptors. And then we can postulate chemical modifications in the target regions of the ligand to improve complementarity. Okay, and these are the methods to determine the structure of the target. Okay, so these three primary methods are useful for the structure determination of the target or the receptor. X-ray crystallography, NMR, and homology modeling. With the completion of the human genome project, we now have primary amino acid sequence of all the potential proteins in a typical human body. But knowledge of the primary sequence alone is not enough for a drug design project. Crystal structures are the most important source of information of drug design, since these structures determined to a high resolution will be available, and the method is useful for proteins that ranges from in the size of a Armstrong to amino acids of many numbers. Okay, three-dimensional structures of these proteins can be determined using 2D NMR experiments and also from X-ray crystallography. And these structures are deposited in PDB, protein data bank. We can directly download these structures from the protein data bank, and we can use for further drug design techniques. If no experimentally determined structure is available, then we go for homology modeling, what we have discussed earlier. Okay, when no structures of the protein is available, we can go for homology modeling. And so now we have structure of the protein or the structure of the target. Now we should have structure of ligands, or we should have ligands. So these are the two methods to design newer ligands. Okay, either we can search in databases for newer ligands, or we can go for de novo design. Okay, and database searching already we have discussed. Many databases are available like Sync database, SubChem database. From these databases, we can select any molecules and we can go for virtual screening. Like that, we can go for drug design. But here, de novo drug design is again a method of design. It means that start afresh, that is from the beginning or from the scratch. We don't have any basic idea of the drug molecule. We are starting from new, completely designing newer compounds. It is a process in which the 3D structure of receptor is used to design newer molecules. Since we know the structure of the receptor, we use that receptor, or we use the shape of that binding site of the receptor for the design of newer molecules. That is, we build compounds that are complementary to a target binding site on a protein by random combination of smaller molecular fragments to make complete molecule with better binding profile. And information available about the target receptor, but there is no existing leads that can interact. In this time, we use or we go for de novo drug design. We know the structure of the target, but there is no molecule which act on the receptor is known. That time we go for de novo drug design. The de novo design methods are fragment placement methods, site point connection methods, fragment connection methods, sequential build-up methods. These are the methods by which we go for de novo drug design. Let's see in short one by one. First of all, fragment placement methods. In fragment placement methods, by the name, from the name itself, we can say it is known as fragment location method. That is, it is a simple approach in which we determine the favorable binding sites for atoms or small fragments in the receptor site. Here, fragments are not connected to form a complete molecule. So, this method is less reliable. It gives only the information regarding what all fragments can be placed in the favorable regions of the binding site of the receptor. And this is the example for that fragment placement method. This is the active site of a protein or the receptor binding site of a protein. We identify the favorable binding regions, and according to that regions, complementary functional group containing fragments are placed in the sites. That's all. That is known as fragment placement method, fragment fitting method. The next method is site point connection method. In site point connection method, it is a point in space where suitable ligand atom can make favorable interaction. The steps involved are site point generation, that is, we search for interacting groups in the active site or binding site of the receptor, and we place small fragments like earlier. We place small fragments in the active site to satisfy one or more interaction points. Then fragments are placed in the favorable regions. Then these fragments are attached, or the building blocks are attached to make contact with the additional site points to build the molecule. I'll show you how it works. In the first step, identification of the interaction sites in the active site or the binding site of the protein. Then fitting molecular fragments. Molecular fragments are fitted in the interaction sites. Then that molecular fragments are connected by linkers, okay, or bridging, fragment bridging. Thus, a molecule is produced. A newer drug molecule is produced by site point connection method. The next is fragment connection method. Here, we place the fragments in the binding site of the receptor, okay. In the binding site itself, we place fragments, and then that fragments are connected in a single step itself using linkers without moving, without rearranging the fragments. That is known as fragment connection method. And the fourth is sequential build-up method. That is, here we sequentially build up the molecule. That is, we construct a ligand either by atom approaches, or by fragment approaches, or by, these are the two methods. That is, we first place an initial core building block in the binding site, and the additional functional groups are added on the initial core. And okay, the additional functional groups are added on the initial core. So in this method, sequential build-up method, we place a core molecule in the binding site, and then additional functional groups which are required for the additional interaction with the target is added to the molecule. That's all. So sequentially building up a newer molecule with more interaction with the receptor. Okay. So that's about the de novo drug design. So now we have come to know the structure of the protein and structure of many ligands. We designed the ligands, we designed protein, we have protein structure. Now we should know whether these ligands are having activity or not. Okay, that is meant by virtual screening. Virtually, we are doing screen. For that is ligand database is there, target protein is there, we do molecular docking. That is, the ligand is placed or the ligand is docked into the protein's active site. So now let us see what is molecular docking. Molecular docking is a computational determination of binding affinity between the molecules, between two molecules. It is a computational determination. Okay, so mostly all the people wrongly assumes that drug design means docking. Okay, docking is not a method of drug design. Okay, there are many methods for drug design. Docking is one of the techniques for drug design. One of the techniques for drug design is docking. Okay, I told you many techniques till now, and now docking is one of the technique of drug design, which is based on structure-based drug design. Okay, which is included in structure-based drug design. And docking actually is the computational determination of binding affinity between a designed molecule and a protein. That is the protein structure and ligand. Docking attempts to find the best matching between two molecules. It includes finding the right key for the lock. We know the lock and key mechanism. Okay, finding the right key for the lock is by docking technique. Okay, so we have seen, I told you, we have seen many techniques for drug design. In that, pharmacophore mapping was there, QACR techniques were there, de novo drug design techniques were there. Now docking techniques are there. Here in docking techniques, we find the right key to open the lock. Okay, so it includes finding the right key for the lock. That is, if two biological molecules are given, docking is used to determine whether these two molecules interact. Two molecules means one is ligand and one is protein. Whether these two molecules interact, if it interacts, what is the orientation that maximizes the interaction while minimizing the total energy of the complex? That is the main aim of the docking technique. That is, two molecules are given, the ligand and the receptor. So when it binds together, whether these two interact, if it interacts, what is the orientation? The orientation in which it maximizes the biological effect, and in that orientation it should have minimum binding energy. Okay, that is determined by using docking. The ligand is docked onto the receptor and the interaction is checked, and find the best orientation of the ligand in active site of the receptor, which would form a complex with overall minimum energy. That is done using molecular docking. And then the result is analyzed by a statistical scoring function, which converts interacting energy into numerical values. And that is called as docking score. The the calculation of energy, interaction energy, and conversion of that energy into numerical values. And that is called docking score calculation. Process of placing molecules in appropriate configuration with the receptor. That is also done in the molecular docking. And after placing this, it predicts whether these two molecules interact or not. And we find out the binding affinity and the 3D structure of the complex. That is a bound complex of the ligand and the receptor, and the optimal orientation and conformation of the interacting molecules in space. Then how stable that complex is. All these are calculated using this docking techniques. And so this molecular docking technique essentially consists of two components. Okay, one is the search algorithm. And next is the scoring function. The search algorithm positions molecules in various locations, orientations, and conformations within the active site of the molecule. Some of the earliest docking programs position a molecule in the active site and then holding it rigid with respect to the conformational changes. But all modern docking algorithms include ligand conformational changes. The scoring function is responsible for determining if the orientation chosen by the search algorithm are the most energetically favorable and is responsible for the computing the binding energy. So these are the two components of the molecular docking technique: search algorithm and scoring function. It is the first binding site identification of the receptor is the first step in the structure-based design or the docking technique, binding site identification. And it relies on the identification of, you can see here, concave surfaces on the protein that can accommodate the drug-sized molecule, and that also possesses appropriate hotspots for interaction of the ligand with the receptor. And hotspots, the hydrophobic surfaces, hydrogen bonding sites that drive the ligand binding. These points interact with these points of the ligand interact with the receptor to have a strong binding with the receptor. Search algorithm involves finding all possible conformations of the receptor and all possible rotational and translational orientations of the ligand relative to the receptor with a given structure. And most of the docking program, I told you, uses flexible ligand, and some use even flexible proteins also, receptor. In binding free energy calculation, this docking scores would be free energy of binding. I told you earlier itself, docking scores means it is the energy of binding. And it is calculated as a sum of electrostatic energy, van der Waals energy, internal energy change due to the flexible deformations, and translational and rotational energy. And we know lesser the binding free energy of a complex, the more stable it is. So the docking score will also be, if it is lesser, the more efficient or the more biologically active the complex will be. This is a total binding energy calculation, that is the delta G bind, that is delta G solvent, delta G conf, delta G interaction, delta G rotational, delta G translational, delta G vibration. All these energies are included in this binding energy calculation to convert it to the scoring function. And this docking can be between what we discussed, protein ligand, and sometimes protein-protein, and sometimes protein-nucleotide docking can also be done, because we know all these compounds, that is drug molecule chemical compounds, proteins, nucleotides, all these are used as drugs. Now, types of docking. All docking programs alter the orientation and conformation of the ligand in order to fit in a protein's active site, which is itself held in a fixed geometry. But some docking programs have the ability to allow the active site of the protein to alter the shape. This results in docking simulations that take much longer time. However, it is necessary to allow the active site flexibility also if the protein exhibits an induced fit mechanism. Even without an induced fit mechanism, it can give somewhat accurate, more accurate results because of the residue side chains being able to reposition slightly. There are several types of induced fit mechanism, and among the simplest is one in which the protein backbone is rigid but one or more protein side chains or residue side chains changes the conformation to alter the position within the active site. So depending upon that, there will be two types of docking: rigid locking, that is, in that rigid locking, the lock and key both are rigid. Only the key can be flexible. The lock is not flexible. Lock is rigid. But in rigid docking, the internal geometry of both the receptor and ligands are treated as rigid, whereas flexible docking or induced fit docking, in that rotations on one of the molecules is performed, that is the key. Every rotation energy is calculated, and later the most optimum pose is selected. This is a typical docking workflow. That is, target is selected. That is, target is, first of all, the structure of the target is identified. Either we select it from PDB or by homology modeling, and then the ligand is selected. Either it is designed, that is by de novo drug design, or it is selected from a database. Then the target is prepared, because the structure of the target will not be complete. It should be pre-processed. Processing should include removal of water molecules from the cavity, stabilizing the charges, filling the missing residues, and the generation of side chains. All these are done in target preparation. And in ligand preparation also, the 2D structure of the molecule will be converted to 3D structure. And these two are given in the software for docking processes. And finally, evaluate the docking result. The docking result will be to the scoring function and the pose evaluation, the pose of the molecule. So, uses of docking, I told you in the initial itself, the docking can be used for virtual screening and also for pose prediction. That is, docking is used for virtual screening to identify potential lead compounds from a large data set, known structures of organic compounds, libraries of chemical compounds, programs which calculate affinity for the protein. So from all these, ligands can be selected for virtual screening using docking techniques. Then pose can be predicted. That is, if we know exactly where and how a known ligand binds, we can see which parts are important for binding. So if we can predict exactly the pose, the binding pose of the ligand with the protein, we can see which parts are important for binding. According to that, we can suggest improvements for improving the affinity. Then we can avoid changes that will clash with the protein. So that's about structure-based drug design. So till in this one and a half hours, we were discussing about drug design techniques. I told you an overall view. I cannot go deeper into this these techniques in this 1 hour. Okay, so we have seen structure-based drug design and ligand-based drug design, when we can use structure-based drug design and ligand-based drug design. And now, if somebody asks which is best, I will recommend we can pursue both receptor and pharmacophore or structure-based approaches independently. If we know structure of the receptor or protein, and if we know the structure of ligand, both is known to us, for a better result, we should go for both. That is, we can pursue both structure-based drug design and ligand-based drug design to give a better result. If the binding mode of the ligand and the target is known, information from each approach can be used to help the other. Ideally, identify a structural model that explains the biological activities of the known small molecules on the basis of their interactions with the 3D structure of the target protein. And finally, we can say the ligand-based drug design or pharmacophore-based drug design is molecular mimicry. That is, we mimic already existing molecules, whereas Receptor-based drug design is molecular complementarity. That is, we know the structure of the protein. In complementary to the structure of that protein, we design newer molecules. So you can decide now which is best. So typical projects are not purely receptor or pharmacophore based. They use combination of information, hopefully synergistically. Okay. So this is the ending of this presentation, but I tell you, every ending is really just a new beginning. So with this short and a brief knowledge of drug design, now you can go deeper into drug design techniques. If anybody has any doubts or if you want more clarification in any of these topics, you can email me or WhatsApp me in this number. Thank you.
Thank you, ma'am, for your nice presentation. Thank you once for all. Thank you.
Thank you, sir.